Welcome to our single-cell multiome analysis series with SciDAP. In the previous video, we were reviewing the output of the Cell Ranger count pipeline, UC matrix, and UMAP view in UCSC browser. Our samples now processed separately and can't be put on a single UMAP just yet. To do that, we need to start another pipeline called Cell Ranger Aggregate for RNA and ATAC-Seq. It will combine the samples in a single feature barcode matrix, so we could rename our new aggregated sample. Let's give it a name and then we select all available samples that belong to this experiment in this drop-down menu. Then we select the index that were built before, and before clicking on Save Sample, switch to the Advanced tab and ensure that the library depth normalization is set to none to prevent subsampling the reads to the smallest dataset. This subsampling may be useful for viewing the results with 10x genomics loop browser, but however, it may lead to data loss. If this option is set to none, normalization and scaling will be performed independently in single-cell RNA-Seq and single-cell ATAC-Seq dimensionality reduction steps later. So click on Save and Run, and wait for the aggregation step to finish running. And in the next video, we will be reviewing the output of this pipeline.